And we're live. Hello, everyone. We just wait a few minutes. Okay, everything seems to be working well. We have the link posted. I'll wait to make sure that everyone can join the Hangouts.
So you posted the link on Twitter, right?
That's right, and then I'll also send it to the SCL membership.
Perfect.
Yep. Hi, Dan. Perfect, we're live. Hi, everyone. This is, I'm Alex, and I'll be hosting this Limno seminar today. I'm very excited to have our first fish talk, and the inaugural talk is by Dr. Katrine Turgeon. Katrine is a postdoctoral fellow at McGill. She's on a MyTax Elevate Fellowship, working with industrial partner, and she'll describe this to us in a minute. Katrine did her undergrad at Sherbrooke in biology. She moved on to do a master's in environmental science at the Université du Québec at Trois-Rivières. She then pursued a PhD in tropical fish ecology at McGill. A first postdoc at Guelph, where she looked and she modeled the socio-ecological complexities of fisheries, and then she moved on to McGill, where she looked at hydroelectric reservoirs and the effect of human activities on fish populations. Katrine, you may try to share your screen.
I'm sharing my screen. Share.
And so just a few comments to those who join us on the Hangout. Just make sure that you mute yourself. And please do not hesitate to stay after the seminar to ask questions. And you can also ask questions directly online at Limno Seminar on Twitter and also on YouTube. The floor is yours.
Perfect. Thank you, Alexandre. So thanks everyone for taking the time to hear what I have to say about fish community dynamics in large reservoir. So what I will present you today are results from my postdoc at McGill University in collaboration with Hydro-Québec as an industrial partner through a MyTax Elevate scholarship. So that's a very valuable collaboration. So in this postdoc, I'm looking at the effect of river impoundment on fish community dynamic using a multi-scale approach. So if you want to learn more about my research interests and what I did, you can have a look at my website. Okay. So one of the most important challenges we face as a society is the increase in energy demand worldwide. So this is not surprising. And in response to this demand, the construction of large dams to provide hydroelectricity experience an unprecedented boom in emerging economy. And this is what the figure represents. So in red, you have the planned hydroelectricity project. And in purple, you have the under construction project. And the size of the triangle represents the storage capacity of the reservoir. So we must really emphasize on the fact that hydroelectricity is presented as being one of the most stable, reliable, clean, and renewable energy sources. However, if we, next slide, if we superimpose the existing dam in orange over the dam that are under construction or planned, we can see that basically only arid or semi-arid region are not affected by the presence of dam. So if we look at some numbers, roughly 49% of main river basins contain large dams, and this number should get very close to 75% in 2030. So this is quite an extensive distribution of them. So since flow is the major driver of ecological processes in river, the regulation and fragmentation caused by dam can strongly impact ecosystem function and biodiversity. So more specifically, dam regulate river flow by transforming large river here, like here you have an ecosystem, a river ecosystem, and they transform a large river into storage reservoir like this one, and create new lentic habitats. Dam also generate variation in water level far beyond natural amplitude in this new ecosystem, because upstream of the dam, the ecosystem experienced some important drawdown, and downstream of the dam, the seasonal and interannual stream flow magnitude and variability are usually and generally reduced. So all those hydrological modification will affect water temperature regime, sedimentation, and nutrient cycling. So it's why we need data upstream and downstream of the dam to really get the influence of impounding a river on the whole ecosystem. So in this figure, you basically have the Paraguay-Paraná River before and after the construction, and you can see that it's basically transforming a large river into a big lake. So what are the impacts on fish community? So these hydrological alteration are suggested to favor the persistence of certain species over others, and this is not surprising. So one of the main problem is dam impede the movement of many migratory species. So this is the fragmentation effect. The problem is the impact of drawdown. So here you have quite an extensive drawdown effect. So drawdown, by changing water level during winter, can strongly impact fish that rely on this littoral bentivore zone. So bentivorous fish are usually more impacted by regulation in a reservoir. And another factor is, it's more a confounding factor, because usually in reservoir, you have an increasing, you increase the susceptibility to non-native species colonization because you have more recreational fishermen coming to the reservoir. So all those factors should ultimately result in a loss of biodiversity. So what is missing to get the big picture? Because we have quite an extensive literature on the effect of impoundment on fish aquatic ecosystem, but they are usually small-scale studies. So I did, at the beginning of this postdoc, I started a synthesis looking at more than 85 studies, small-scale studies looking at the impact on the population and community level. And many of them basically suffer from very few replicates, and this is what is illustrated on your right. So most of the study use only one reservoir, and another shortcoming is the need for control site and reference sites, because if you want to follow reservoir trajectory over time, over several years, you need a kind of a reference site that is not impacted to really see what's going on and to account for climatic change or stochasticity. Another problem is the time series are usually very short, so reservoir follow for just a couple of years, or usually just 1 year, and it's a snapshot in time and space. So there is a couple of shortcomings in those small-scale studies. We also have large-scale studies, but those large-scale studies usually superimpose different maps together, fish diversity map and dam maps, and try to make correlation, but they do not really measure the single effect of impoundment on fish communities, so that's another shortcoming. And usually we have very little information on the mechanism. So we need a kind of a synthesis and a general effort to really understand the multi-scale effect, temporal and spatial, the effect of impoundment and water regulation on fish community. And it's what my project is about. And another challenge when we want to evaluate the impact of human activity on ecosystem is to deal adequately with scales. Because the scales selected should be of ecological relevance to account for, let's say, species assemblage and their interaction with the environment. So we really need to make spatial, we need to put special care in the examination of several spatial scales. So in this project, I look at two main scales, one global, using literature review and synthesis. So I took several data from around the globe and try to extract some generality. So my pattern are general. But I also had a unique opportunity to work with Hydro-Québec and have a very detailed data set at a more local scale. So it's really in the Quebec boreal region. But these two scales are complementary to really understand the impact of impoundment on fish community. So let's look at my local scale in more detail. So first, I call it my local scale, but the La Grande Rivière complex in northern Quebec, so it's basically located here. This is roughly the size of New Brunswick. So it's local, but pretty large. And this local scale also consists of different smaller spatial scales. So we can look at the whole hydroelectricity complex by combining all the data set from the hydroelectricity complex. We can also look at the reservoir scale. So looking at the station, so combining the sampling station of one reservoir and looking at the 3 different reservoirs. So each little dot here represents sampling station. And we can also go at a finer scale and really try to understand what's happening at each little sampling station in those reservoirs. So here you have the St. Marguerite River complex. So I also have data on that, but I will really focus on the La Grande complex today just for simplicity. So this fish community data set collected by Hydro-Québec is quite unique, and I think it's allowing us to do the most thorough evaluation of impoundment on fish community. And it's unique in many respects because first we have data before impoundment on several sampling stations. So here on the picture you have what the Robert Bourassa Reservoir looked like before impoundment, and each little dot represents small sampling station. So in pink you have stations that are upstream of the dam. In blue, you have stations that are downstream of the dam, and each reservoir was paired with a lake to really get a reference size. So this is before impoundment, and this is what the reservoir looked like after impoundment. And just to give you an idea of the size, this is the island of Montreal. So those reservoirs are quite huge. So as I told you, we have upstream station, we have reference station and downstream station. And another complexity of this data set is, let me just back up. So we have stations that were river before and stations that were lake before. So we can really get an idea of what is the impact of impoundment on fish assemblage that were in a river before and in a lake. So one might expect that fish community in a river experience a more drastic change than being in a lake and transforming into basically a bigger lake. So this complexity is also, I think, quite important. So this data set is also quite unique because we have a standardized fish sampling in all the stations. We have roughly 20 years of data after impoundment. And I think maybe the most unique characteristic of this data set, we have almost no confounding factors. So there is no land use, there is no fishing intensity, there is no invasive species, there is no stocking. So we can really measure the single effect of impoundment on fish community, which is quite amazing. And we also have water quality data to go with fish assemblage over time to really see if a change in fish is related to variation in water quality data. So how do we measure fish response? to impoundment. So here is another scale. It's the scale of biological complexity. So one might be interested to look at the effect of impoundment on fish growth and condition. So even though this is probably very interesting, I will not cover this today, and this is work in progress. So it will be covered in my project, but I will not cover this today. So today I will focus on the effect of impoundment on fish population level and on fish diversity and community level. So let's start with what's happening at the population level. So to see what is happening at the population level over time following the impoundment of the river, I decided to revisit the trophic surge hypothesis. So this hypothesis makes prediction about how the increase in phosphorus loading from leaching and from the leaching and decomposition of organic matter from newly flooded terrestrial area would affect productivity in reservoir. So on the figure, you have basically the system before impoundment. So it was a river. Then you increase, so you basically fill the reservoir and you get towards a dynamic, regulated dynamic in the reservoir. So how this should be, how this can be translated biologically. So during the surge, so we should have a large influx of allochthonous inorganic nutrient and organic detritus from the flooded area, and this should translate quite quickly into a high primary production and a high bacteria production. Flooding the terrestrial area also result in an expansion of a lot of habitat, and this will create new ecological niche, and coupled with high primary production, this should roughly result in a very high productivity of zooplankton and benthas. This should be followed by higher fish recruitment that will translate into a higher adult fish abundance over time. So this is basically what the trophic surge hypothesis is about. So we have roughly 3 phases. So we have the surge, which is, so I took the blue line for now to put my different phase. So you have the surge, where you have an increase in abundance in fish, as an example, and you have a depression phase, where the available nutrient and detritus stocks are exhausted, and this should basically bring down all the different trophic components down as well. And we have finally the trophic equilibrium, where the reservoir should stabilize toward a kind of a steady lake-type ecosystem. So one of our goals was really to test if this hump-shaped pattern, so the trophic surge, is really prevalent in some time series available from the literature. So we collected, so we did some data hunting and found roughly 40 recruitment time series and 109 adult fish time series that came from 7 different reservoirs in boreal and temperate ecosystem. I unfortunately did not found any time series from tropical ecosystem, but the comparison of the 3 ecoregion would be fantastic. So to test if the trophic surge hypothesis was supported by each individual time series, we did some curve fitting and compare the fit of the data to different alternative scenario. So we compare the no pattern scenario, so basically a flat line, some increasing trend, some decreasing trend, and some hump-shaped pattern. So if we actually detect a trophic surge, we should have statistical support for a hump-shaped pattern. So here you have the result for recruits and adults. So on the y-axis, you have the number of time series that that we detect statistically an increasing trend, a hump shape, a decrease, or a no trend. So here are the results. So roughly 43% of the time series on recruits show a statistical hump-shaped pattern. And we have a very similar pattern for adults. So we have quite a good support for the trophic surge hypothesis when examining individual time series on recruits and adult fish. What about when we combine the data set together? We actually have a mild support for the trophic surge when we when we combine together all the time series. We found no support. So if we look at the recruits here, we basically have no support when we combine boreal and temperate ecosystem together. So it's the black line. And we do not have any support in temperate, but quite a good support in boreal ecosystem. For adult fish, we have a very mild support when we combine data set together, the black line. So support for trophic surge, so a hump shape, and and some support for a hump shape in adult and boreal reservoir, but it's more a decreasing trend in temperate reservoir. So these results suggest that there's probably many confounding factors that mask the detection of the surge when we combine together all the data set. And this is not very surprising because many factors can influence the magnitude and the detection of the surge. So we test for ecoregion. So one might expect that in boreal region where temperature is lower, we might see a longer trophic surge. So everything takes a longer time to stabilize. Where in tropical, maybe the trophic surge is happening very fast and then stabilize toward a new ecosystem. Another factor that could potentially affect our ability to detect the surge and affect also the magnitude is the filling time. Because if it takes you 7 or 9 years to fill your reservoir, maybe the trophic surge will be quite longer because the nutrient in the water will be released at a slower rate, and so the trophic surge could be slower, but also of lower magnitude. And well, there is other characteristic of the reservoir that can also affect the detection and magnitude of the surge. So the size of your reservoir and the residency time, and obviously different species will react differently. So all these factors can basically explain variation in the different the magnitude of the surge and on our ability to actually detect the surge. So to go a step further and understand what caused variability in our ability to detect the surge and what caused difference in duration and magnitude, we extract 5 trophic surge metrics that could be used in a predictive framework to really see what's going on. And these metrics were first, if we detect or not a trophic surge, so detection of a hump shape or not, the duration of the trophic non-equilibrium, so starting from filling to basically where we reach the new trophic equilibrium. We can also extract the duration of the trophic surge, so from the start to the peak. We can also detect the magnitude of the trophic surge, so comparing abundance value before and at the peak. And we can also extract information on the timing of the peak, so where we get the highest magnitude. So we did that. For each individual time series we had, we basically extract all those metrics and trying to come up with some generalities. So here you have 2 examples of trophic surge metrics that we extracted. On the panel A, you have the duration of the trophic non-equilibrium, and here you have the duration of the surge on panel B. The blue line here, the bigger, like the bold blue line, are adult fish in boreal reservoir. Here you have recruits in boreal reservoir. The green line are adult fish in a temperate reservoir, and the dash line, green dash line here, are basically recruits in temperate reservoir. So as a first observation, you can see that there is quite a lot of variation among the time series and among the different region. The trophic non-equilibrium phase seems to last about 9 years in recruits for both regions, but can last, I should say, at least 14 years in adult fish in boreal ecosystem. And this is a very conservative estimate because many time series did not convincingly reach the equilibrium phase at the end of the data set. So I think this is probably closer to 15 and even maybe like 16 or 17 year because it's very, very conservative. We have a little bit more confidence in the duration of the surge because almost all time series covered the duration of the surge. So the surge lasts about four years in recruits and about 5 years in adult. And this is, so if we put duration of the surge in relation to the duration of the trophic non-equilibrium, this is suggesting that the increase, so the surge, is probably more faster than the depression phase. That probably took a longer time. And we also try to correlate those metrics to some reservoir characteristic, but with only 7 reservoir. I mean, our statistical support was, our power was quite low, but it looks like the total surface of flooded area explain almost everything. So the bigger your reservoir is, the longer your surge, and the longer the duration of non-trophic equilibrium is as well. So we've been through the population level. Let's jump to the diversity and community level, because when you want to measure fish response to impoundment, you need, I would say, at least 2 scale of biological complexity, population and diversity. So how do we evaluate the effect of impoundment on diversity and community? So first we can, and I should mention, so the next 2 slides are from this review and synthesis, so they are at the global scale. So one way to evaluate the effect of impoundment on fish diversity is to look at its change over time. So we have data on a reservoir and look at what is happening before, during filling, and then after. So here I have to admit that maybe that's not the best graph, because here I have like roughly 60 mini time series, and putting them separately on the graph, it just looked like a big spaghetti mess. So I combined them together in terms of their ecoregion. So in blue you have boreal, in green you have temperate, and in yellow you have tropical. On the x-axis is basically time since impoundment, and on the y-axis is the difference in number of species before and after. So if we have sensibly the same number, we should be very close to 0. If we are losing species, we're going to be down 0, and if we are gaining species, we're going to be here in the upper region. So one conclusion is we don't have any drastic loss of richness of species in boreal and temperate ecosystem, but there is a little bit more variation in tropical ecosystem. And here we have really, you might help me on that, because I'm still like struggling a little bit in how I should analyze that, because we definitely have a quite serious scaling issue here, because is losing 1 species in boreal corresponds to losing 20 species in tropical. So there is, it's like comparing orange and apples. And if we standardize everything, I try to standardize the data set, and losing half of a species in boreal corresponds to losing 20 species in tropical. So can they be comparable? I think it's a question we should ask ourselves, and I will be happy to take any comments and to discuss about that. But in general, globally, tropical ecosystems seems to be more impacted. Another way to look at the impact of impoundment on fish diversity is to compare what's happening in the reservoir versus what's happening in reference sites. And from the literature, we usually do not have time series to look at that. We have a snapshot in time, looking at diversity in a reservoir versus diversity in reference. And reference sites can be lakes or rivers. So to do that, I did a meta-analysis, which is to make a long story short, is a statistical analysis that combines the result of multiple scientific studies together into a forest graph like that. So a value of 0 means that you don't have a difference between reference ecosystem and reservoir. If your study fall on this side, on the right side, it means that you have a higher richness in regulated ecosystem, like in reservoir or regulated stream. If you fall on the other side, it means that you have a higher richness in reference side. So let's just plot one study. Study 1 compare reservoir versus lake in the boreal ecosystem. So you can see, and when the whiskers, so when the bar line cross the 0 line, it means that it's not statistically significant. So this is one study suggesting that the trend is a higher richness in regulated ecosystem. And if we put together all the study, so in dark blue you have all the study that look at lake versus reservoir, and in light blue you have the study that look at river versus a regulated ecosystem. So the global effect cross the red line. So we do not have a significant lower or higher richness in reservoir reference ecosystem. Some river has actually a higher richness in regulated ecosystem. And if we look at where they were, in terms of like boreal in blue, temperate in green, and tropical in yellow, there's not a clear pattern either. And I'm still populating this data set, so I'm hoping to find a couple of more study to really populate that, because we don't have a lot of good study on tropical ecosystem, and I think it's where we're gonna actually detect a loss in richness. Like here, this is a statistically significant lower richness in regulated ecosystems, so a higher richness in reference. So globally, no difference between regulated and non-regulated site. So a much stronger approach would be to look at spatial temporal patterns. So basically comparing change over time in impacted station and reference sites. And this is what my local scale, like data from Hydro-Quebec's allow me. So just a quick reminder of my 3 scale. So I have data on the complex level, combining all the data together. I have data, I can combine data per reservoir. So I have 3 reservoirs. And I also have 28 individual station, upstream, downstream, and reference sites. So here, let's look at classic diversity metrics, total richness. In green, you have, and this is at the complex level, so combining everything together. So you have reference sites in green, impacted station in black. And I put the points like every single dots here just to show you the magnitude of the variation, but for simplicity and clarity, I will just remove them. So when we compare, so here what we need to do is really compare the slope, so the rate of change over time. And when we compare reference sites with impacted station, the difference is not significant. Even though I see a little decrease, a stronger decrease in impacted station, this is not significant. And as I told you earlier, maybe the different station, the impacted station matters. So here are the dots again. I will just mask them for clarity. So the green line here represents the reference sites. The blue line represents the station that were downstream of the dam. And as you can recall, I had 2 upstream stations. One, some that were lake before, like the light orange, and some that were river before. So again, even though the trend seems to be important in upstream station that were river before, the trend is not significantly different from the reference sites. It might be a power effect, but we have so we have quite a lot of dots. So we have very similar pattern for diversity and evenness. These are 2 other metrics, classic diversity metrics. And the variation is even smaller. So the diversity and evenness line are closer to the reference sites. And we also have very similar pattern when we look at complex, reservoir, and station level. So for simplicity, I will just show the complex level, but the scaling effect, so upscaling and downscaling effects works very well when we look at classic diversity metrics. Okay, so we haven't found any impacts on diversity, but can we detect impacts of impoundment on fish assemblages? Because we are transforming a big river into a big lake, so it should, in theory, impact fish assemblages. So to do that, I run some RDA, so redundancy analysis. They are ordination techniques. And I also use variation partitioning to really understand what's going on at the assemblage level. So the beauty of using variation partitioning is that I can constrain a change in assemblage with different matrix. So one matrix that is of interest is basically time since impoundment, because we really want to see if fish assemblage change over time. Another matrix that should be important, and this is important at the complex and reservoir level, C and R, is the spatial heterogeneity among my station. So here I just enter the name of the station to control for spatial heterogeneity. Another matrix that should be important is the water quality variable. And a third one, just to control for the fishing gear, is the fishing gear, because we use they use 3 different nets, and this should bring some variation. So I just control for it in the analysis. And so the beauty of variation partitioning, we can get information on shared variation between different matrix, which is quite useful, because let's say fish assemblage change over time, but it's tracking environmental, like water quality. We should get a higher variation explained by this particular area here. And there is no, in variation partitioning, even though some variables are collinear together, it's not a problem, because it will be taken into account in this shared variation section. So, and we are particularly interested to see if time affects species assemblage. So what's happening at the station scale? So here I have 28 in this one, because Sainte Marguerite is not included, so sorry, so N should be 28. So most of the variation is explained by the share effect of time since impoundment in red and water quality in orange yellow. And this is exactly what we would expect. And this is an example of what happened in one sampling station. And here, this is an average of 24. So this is the result from all the stations. So there is some standard deviation associated with this value, but for simplicity, I just put the average. So most of the variation is explained by the share effect of time and water quality variable. And here's an example of how the population, the community change over time. So before the dashed line, you have what was there before impoundment and what was there after. The name, this is basically the name of the species, and they are code to represent the genus, the first 2 letters of the genus and the first 2 letters of the species. So what we can see, we are losing walleye in purple. We are also losing white sucker, and we are gaining quite a lot of pikes. And we are also gaining white fish, so corygonids. And this seems to stabilize roughly 5 years after impoundment. And this is a consistent trend we can see in many, many upstream stations. So what if we look at change in species assemblage at the hydroelectricity complex and reservoir complex? When we look at those higher, bigger scale, spatial heterogeneity explain almost all the variation in species assemblage shared with water quality. So time since impoundment explain a much lower variation. And this is not very surprising, and this is why it's so important to look at different scale when we are examining species assemblage. Because why we only detect the effect of impoundment at the sampling station? It's because at the complex and reservoir scales, the spatial heterogeneity among my sampling station and reservoir basically mask the effect of impoundment on fish assemblage. Just because at those scale, it's the ecological filter that really drives species assemblage. And it's about how geography and isolation created quite unique population community in those different reservoirs. So we are really capturing different processes. So it's why looking at various spatial scale is so important. So maybe someone want to know what's the effect on different species. So which species are affected and in which direction. So if we use my spider web graph here, I plot the 11 most common species for simplicity. And I use some beta diversity analysis, but just for simplicity, let's say that we can really see which species is the mostly vary the most. So which species has the higher turnover rate over time by looking at those graphs. So let's plot the upstream station effect. So we can see that in upstream station, the two catastamids seems to be affected. The two carigonids are also affected, so it means that there is a higher turnover. The pike is also strongly affected, and the walleye is also affected in upstream station. But we don't have the direction now, it's just how much variation there is for a given species. If we look at the downstream station effect, other species are more affected. So there is difference between downstream and upstream station, but we have quite a lot of variation in reference station as well. And this is mainly due because we have quite a lot of variation between reservoir as well. So some species are more affected in Opinyaka versus Rababourassa. So the best way to look at that is to isolate the pattern per reservoir. So here you have, so to really understand what's going on, we're going to look at Rababourassa, and we have quite similar pattern in Opinyaka and Kanya-Pisco, like the two other big reservoirs. And when we look at each reservoir separately, we can have the direction of the change here with those little bar graphs. So we can really see which species is affected. So you basically have like 5 upstream station, 1 downstream, and 1 reference sites in Rababourassa. So what we can see, we observe a general change from a catastamid dominated population community towards a pike corygonid. So white sucker decreased quite drastically in upstream station, and we are gaining cisco, whitefish, and we are gaining a lot of pike. And this is totally consistent with the graph I showed you earlier. So we have this, and this pattern is quite consistent in Rababourassa and Opinyaka. So some species are detrimentally affected, the walleye as well, but some species benefit from impoundment, like the pike and the whitefish. And the First Nation are quite happy in the Bay James because those fish are, the cisco and the whitefish are very important for native, for First Nation. So key findings of this project. So regarding the trophic surge hypothesis, we have quite a good support for the hypothesis when we look at individual time series, but milder support when we combine them. And the non-equilibrium phase can be quite long, so we have to be careful with that. At the diversity and species assemblage level, there is not a drastic loss in biodiversity in boreal and temperate ecosystem, but this seems to be apparent loss of richness in a tropical system. We also observe a change in fish assemblage, which is not surprising. We are transforming a large river into a large lake. And I will really emphasize the importance of validating the upscaling and downscaling effect, because we found only an impoundment effect at the station level. So if we need to make recommendation, we have to be very careful to develop management recommendation for fish before reaching the trophic equilibrium, because those formulation can be short-sighted. And the next step is really to explore this trophic surge hypothesis, but using the data from Hydro-Quebec. So I will reduce quite drastically the variation, and I can really focus on the species effects. So how the different species react to trophic surge in different way, in different manners. So this will be quite interesting. Regarding diversity and species assemblage, doing a multi-scale approach is quite critical in evaluating the effect of hydroelectricity production on fish, and probably on human impact on many ecosystems. And I also have concern about using richness as a measure of anthropogenic impact on fish community, because doesn't seem to be quite, the richness doesn't seem to change, but it considered very little mechanism, because if we have invasive species, we have an increase in richness. So we should really probably break down richness into different components to really understand what's going on. And I want to thank all those people. A lot of very fruitful discussion in the Solomon and Gregory Eves lab. And I really want to thank the biologists and scientists from Hydro-Quebec, because they give me very cool advice and guidance. And some other member of a group I'm part of, and all the founding support and agencies. So I guess I will stop sharing now. Okay.
And we're back.
We're back.
Thanks, Katrine. Very interesting.
Thank you.
I need to work on getting to this better, because I couldn't tweet everything properly. So my apologies to all the fish-inclined scientists out there. The floor is open for questions. You may ask your questions directly on the Hangout here. Make sure you unmute yourself and mute yourself back once you're done. You can also ask questions directly on Twitter at Limno Seminars. And you can also ask your questions in the comment section of YouTube. Go ahead. Jérôme, you have to unmute yourself. I see your lips working, but nothing gets through.
It's not working.
No.
So maybe you can write the question.
Can you try to write the question in the comment section of YouTube or? No. Maybe I can try to unmute yourself. No. Sorry.
So Jérôme, do you want to maybe write your question to Alexandre, and maybe I can answer it? Okay, sweet.
So I do have a question. As you know, there've been quite a bit of we've heard quite a bit about impoundments on the news lately with respect to mercury contamination in Labrador, in Labrador and Newfoundland. And so this is really interesting how there is this succession, I mean, probably expected, but of different fish species after the impoundment is set up, and these changes over time that you identify. And I'm wondering if you're aware of studies that have actually looked at contaminant transfers, or if modeling efforts have tried to look at potential transfer, trophic transfer of contaminants as those food webs are changing through time.
Yeah, there is a team at Hydro-Québec that is actually addressing that. It was addressed more in the 80s and 90s, but Roger Chetang is the biologist at Hydro-Québec, and he did a very amazing. He wrote a couple of very good papers on that, and the interesting story is the mercury level really followed kind of the trophic surge pattern.
Right.
So it seems to last roughly 20 years, I think, in pikes. So pikes are the most affected species because they are really at the top of the trophic web. And apparently after 20 years in the Bay James region, the mercury is not toxic anymore.
Right.
So you can eat pike without any.
That some of the First Nation people see this as those top predators becoming more abundant as something very positive, but there might be a message also with respect to what is the window of time where it's safe to consume there, isn't it?
It's exactly why Hydro-Québec did this study, because the First Nation up there, they rely so much on fishing and hunting. So it's why they established some norms to really try to reduce the consumption of fish that were high in mercury, because all those bacteria apparently release mercury, and it's going through the trophic web like quite fast, and it's lasting quite a long period of time, like 15 to 20 years is not trivial.
Not at all.
But after that, it's coming back to level like before impoundment. So
good, thank you.
You're welcome.
So I'm still trying to get a few questions. I'm trying to scout. I can't seem to see your question, Jerome, if you're trying to send it again. Oh, perfect. So there is a new question. So we have a question from Roberto Q. Hi, Roberto. So here it is. I believe you mentioned once about statistical power possibly being an issue in trends. If there was data available, are there additional systems in particular you'd like to add to analysis?
Regarding the diversity, like so he probably refers to the diversity I have with the Hydro-Québec data. So it will be quite difficult. So I need more reservoir that are tracked. So I need basically more reservoir that are tracked over time on such a long time period, which is almost impossible to get. So in the best worst scenario, I could put together and combine together several reservoir from all over the world and really look what's going on. But this data set is, I think, the best I can get at the moment with such a good spatial extent and temporal time series. So it's I think this trend. I think that the trend is valid. I don't think we have a strong impact on richness, evenness, and diversity. But as I said earlier, are they good measurements to really evaluate the impact of impoundment on fish diversity? So in the boreal ecosystem, I think they are because we have very little confounding factors. So we don't have any invasive species, we don't have stocking, we have a couple of mitigation measures, but it's not adding species so it's not affecting some species drastically. So I think in my system, in the Hydro-Québec boreal region, it's still a valid problem, and the power is maybe not that much of a problem for this ecosystem.
Thank you.
Yeah.
Roberto, don't hesitate to post a follow-up question if you have any other comments, I'll make sure to ask you questions. Anyone else?
Jérôme is still trying.
Sure. Yeah, Roberto says thanks. Good. Jérôme, another attempt. You can write it. You can definitely send me an email, or use Twitter, or use the comment section on YouTube. You might also write it on a piece of paper, and I can try it.
And just show it in.
The old-fashioned way. So how long is will you be doing more field work this year, this coming field season?
If I'm doing field work?
No.
Is it your question?
Yes.
Yeah, I'm no field work for me. And this data set are from the 78 to 2000. So they are not collecting data since 2002, I think, on the Lagrange complex. And I would be very curious to see what's going on right now, just to see if the system basically reached the trophic equilibrium, and if the species assemblages is still progressing, because it seems to be quite stable after 5 to 6 years. But we never know what's we are quite, well, 20 years is fantastic, but it was basically just reaching the trophic equilibrium after 20 years. For many.
Well, I mean, that's why long monitoring is so important, right? Beyond 20 years.
Yeah, but it's just a question of like money as well. So they need to answer government concerns, and after 20 years, the government was pretty happy with not asking for additional sampling.
We have a few more minutes.
I don't know if there is a problem, like many faces are frozen.
See if I'm getting an email from Jérôme, and then otherwise I'll just cut the broadcast.
Okay, I can see, so I think Jérôme is, I can maybe try to open a group chat. Yes.
Can you see that?
I can see that.
Good. I should probably try to find some sort of a little ambiance music while we're waiting for questions. You know,
okay.
Our experience.
Question. The impact of nutrient is fairly low for boreal system where nutrients are so limiting. Why would this be? The impact of nutrient is fairly low for the boreal system where nutrients are so limiting. Where nutrients are not that, I'm not sure to really get your questions. You mean, I would expect more of the variation related to nutrients. You mean the phosphorus, the big phosphorus surge? Only 7% is nutrient related. I'm not exactly sure what you are referring to, Jérôme. So,
The partitioning of variance.
Oh, so you mean
you had time since impoundment, water quality,
The water,
27, 20.
matrix. Let me I will maybe try to go back so you will not see that, but this will help me to explain. So water quality. So water quality for me is not just nutrients, and there is actually almost no nutrient statistical evidence that nutrient affect fish assemblages in my system. It seems to be the transparency of the water. It seems to be the transparency of the water and the dissolved oxygen that seems to explain fish. But the data on nutrients are not that good. So it was hard to really find good nutrient data collected from Hydro-Québec over time and in space. So sometimes there's just missing data. But there's maybe a way of like digging a little bit more and try to find better data. But the water quality that were significant in my system was dissolved oxygen was the best one, and water transparency, probably related to this change in pikes and walleye that are predators and rely quite heavily on transparency to be successful in their capture, their hunting rate. Oh, and Jérôme is asking the question if the variance explained could be, if my fish assemblage could be explained by Mercury. Actually, Mercury doesn't affect fish at all. So there's a study just published recently from Hydro-Québec people, like in 2015 in the CGFS, and actually it's not affecting fish at all. It's more human. The impact is more on human, so human consumption than actually on fish. You
mean there've been no evidence of sublethal behavioral effect?
No, and they look at growth, they look at body condition, they look at. I think that was the two main factor, and there is absolutely no evidence that mercury strongly has an impact on fish population and community.
Okay.
And there is, so Jérôme told me that there is data on GHG. Yeah, so there's another group in Hydro-Québec working on this impact. And I mean, I think my dream will be to build a team. I need geobiochemists, I need limnologists, strong limnologists, to really try to come up with a conceptual framework about this trophic, starting with the trophic surge hypothesis, and really try to apply it to very, very different trophic level and other, apparently the GHG followed relatively similar pattern, a kind of a hump-shaped pattern. So we have mercury that follow that, we have GHG that follow that, we have fish community that follow that, probably phytoplankton, zooplankton, but at a much shorter, a spike that should be much shorter than fish. So it would be very cool to come up with a very general framework looking at all those different aspects of novel ecosystem, because this is what reservoirs are. They are basically novel ecosystem, and just to really understand how and when they reach a new trophic equilibrium is, I think, very important and would be a very cool multidisciplinary project.
Good. And Joe mentions that there is a book available looking at greenhouse gases impact on pollutants. It was published early in 2000. And if you need access or reference to this book or other literature, do not hesitate to tweet us at Limnos Seminar. So Katrine and everyone, thanks for your participation. We'll meet again in a couple of weeks, 3 weeks, I believe, with Dr. Andrea Kirkwood, who will be talking to us about urban stream ecology. So thanks, Katrine. Stay tuned. Oh, go ahead.
Danielle just has to ask a very quick question, asking, are there any studies about what happened to the fish community once the dam ceased operation or is taken down? Not a lot, because they are starting to take taking down dams. So, and Hydro-Québec, I think they took down 2 or 3 dams recently, and they're just starting to follow what's going on. So it's a new hot topic that will probably be very popular in the following years, because you are basically retransforming a large lake into a river. So it's impacting. It's totally a new ecosystem as well. So, but there's not a lot. Yeah, and Jérôme, tell that dam removal is way more common in the US. Yeah.
Those are a very, very good point, with lots of possible impacts on the fish community, obviously. Okay. Well, thanks everyone. We have to stop the broadcast. Thanks, Katrine.
Bye bye.
Bye everyone. Bye.
Cheers.